So, hello and welcome to today's varvis webinar about the IVDR and its impact on clinical diagnostics. The next round about 60 minutes, we will discuss how the IVDR will turn laboratory diagnostics in Europe upside down. Before we start, let me briefly introduce to you who we are as we are hosting this webinar today. My name is Yvonne Kassmann. I'm product manager here at Limbus. Limbus is located in the north of Germany, right at the Baltic Sea. We are a software development company, a young team and medical device manufacturer, which means that we develop and operate our products according to international standards. Our product varvis is a cloud-based clinical decision support system and a complete solution for all clinical NGS applications, from targeted NGS panels to whole exome sequencing up to cancer diagnostics. varvis is a CE-labeled medical device specifically made to aid in the diagnosis of patients. So we decided to host this webinar today because in the last months and weeks, we, in discussion with many people working in the field of clinical diagnostics, with partners and customers, we learned that there's so many open questions regarding the IVDR and its applications for the clinical routine. So this is why we're very happy that we really have 3 experts here in the room today to share their expertise and to answer all your questions. So the first part will be about how the IVDR raises the bar for diagnostics laboratories across Europe, presented by Dr. Sebastian Grumminger from the Jona Institute. The second part is presented by Dr. Ben Liesfeld from Limbus Medical Technologies, how to deal with software in clinical diagnostics. And the third part is about performance evaluation, where Björn Novak from SenseID will explain what this means and how to deal with it. We will end with hopefully a very lively Q&A session. So if you have questions, please let us know. You can type in your questions depending on the application you're using. This question mark icon should be either in the menu on the left of the application, or if you're using a mobile device, this should be located in the header. If you want to address your question to a specific speaker, you can also indicate this when sending us your question. The first question that we already received is if this webinar is going to be recorded and made available. Yes, this is recorded and we make this available later. So let's get started with the first presentation, how the IVDR raises the bar for diagnostic laboratories across Europe, especially with respect to in-house developed tests. For this, I would like to welcome and introduce to you all Dr. Sebastian Grumminger. Hi, Sebastian. Great to have you here today.
Nice to meet you.
Sebastian is a molecular biologist by training and an expert in the field of in vitro diagnostics. After having obtained his doctorate in the field of virus-associated tumor development at the Helmholtz Center in Munich, he played a key role in the development, practical implementation, approval, and performance evaluation of non-invasive prenatal testing methods. And with this experience in the development and approval processes, he basically took over quality management and regulatory affairs in this company since 2017. He's a strategy expert and team lead for IVD at the Jona Institute, which offers a broad range of services, which we also use quite frequently. So, how the IVDR raises the bar for diagnostic laboratories across Europe. I'm handing over the slides to you and enjoy.
Thank you very much, Yvonne, for this kind introduction, and I'm very happy to be able to present here today, and thankful for inviting me and providing me the opportunity. So yeah, my job today is to give you some insights what the IVDR, how the IVDR impacts on diagnostic laboratories, and why this is now a European topic, not only a topic within the member states. Yeah, so I'm trying to take over the presentation. Yes, it works. So you may feel like in such a situation where the legal system shoots with bullets on you, and we feel the same. The number of requirements and the number of laws, the number of standards is quite larger than those few bullets here, but I selected a few that are pretty relevant in this context, which may be standards like ISO 15189 for laboratory, medical laboratory quality management systems, ISO 17025 for laboratories without a medical context, national laws, in vitro diagnostic medical device directive, which is the current legislation, the IVDR, which is the future regulation. Then we have standards for manufacturing of medical devices. We have standards of life cycle and IT security, a software lifecycle and IT security, which are applicable. So to demonstrate to you how the legal framework is set up in the European Union, we first have a look at the situation. Currently, the EU directives, the Medical Device Directive, Active Implantable Medical Device Directive, and the IVD Directive do provide how to set up the national laws for the national legal bodies. So the national laws and regulations are then applied to citizens and companies. And this is changing dramatically currently, which we have already learned with the GDPR recently, where EU regulations directly impact on citizens and companies. And that is the situation we are facing for the IVDR and the MDR in an equivalent way. So the situation is as such that the Medical Device Directive and the Active Implantable Medical Device Directive are fused into the MDR, and the MDR and the IVDR came into force in 2017. But the difference is the IVD directive is replaced by IVDR, but the changes that come along with the IVDR are much more dramatic as compared to the changes from medical device directives to MDR. It was supposed that MDR should be applied in May this year, so in just a few days. It will be applied now in 2021 because it was postponed due to the Corona pandemic situation, and this was due to the fact that the audits could not be performed on site. This was one of the reasons why the MDR was postponed to this year. However, there is not yet any postponement of the IVDR foreseen right now. So we still think that the IVDR will be applied in May next year. So there's only 1 year to go until the IVDR applies. So we look particularly into this situation, and before we can even say a device is an IVD, we first of all need to find out whether it's a medical device. So therefore I need to bore you with this definition of a medical device. So a medical device can be anything. It can be an instrument, an apparatus, appliance, software, reagent, implant, material, or any other article in the case that it is intended by the manufacturer to be used for a medical purpose. This can be alone or in combination, and it must be for human beings. If this all applies, then you need to look into those bullet points, whether your product does something that applies to one of these points, whether it does diagnosis, is intended to deliver a diagnosis, prevention, monitoring, prediction, prognosis, treatment, or alleviation of a disease. Pretty much the same is true for any disability. the investigation, replacement, or modification of the autonomy, and a physiological or pathological process or state is investigated. This is very much the case for any in vitro diagnostic product. And now it comes very concrete, providing information by means of in vitro examination of specimen derived from the human body. And this is particularly true for any in vitro diagnostic medical device. So we see that every IVD is a medical device per se, but then the MDR states in the case that your product fulfills the definition of an IVD according to IVDR, then IVDR applies and not MDR. So therefore we need to look into the definition of an IVD. And here you see an IVD is always a medical device, which is a reagent product, a calibrator, control material, kit, instrument, apparatus, piece of equipment, software, or system. So you see indicated in red here is the tiny term software, which was changed from the IVD directive to the IVDR. So we have software within the scope of application of the IVDR, and this changes dramatically the situation for decision support systems like varvis and other software that is a standalone software used to perform diagnosis based on data that is derived from specimen analyzed in a laboratory. Whether used alone or in combination, intended by the manufacturer, whether used alone or in combination, intended by the manufacturer to be used for the in vitro examination of specimen, including blood and tissue donation derived from the human body, providing information for one or more of the following medical purposes. And here you see again concerning a physiological or pathological process or state, concerning a congenital physical or mental impairment, and concerning the predisposition of a medical condition or disease. You see here the predisposition is within the scope now indicated in red. To determine the safety and compatibility of potential recipients was always the case. This is the blood donation and tissue transplantation topic. And now we see that to predict treatment response or reactions is the CDX, the companion diagnostics scope, which is now within the definition. And then to define monitoring treatment measures is as well a medical purpose covered by the IVD definition. If your product is intended to perform such actions and provide these statements towards the patient, towards physician for the benefit of a single patient, then it is an IVD medical device. So we can simply say IVDs are medical devices. So IVDs are a subgroup of the entirety of medical devices, which gain medical information obtained from human specimen. And this is very much true now as well for software. So the intended purpose is crucial to decide whether it's an IVD or not. So this is the case, and as well for the classification of the product, to analyze risks that are associated with the device and to meet regulatory requirements. And if we look into the requirements for laboratories, you see that the medical laboratory standard ISO 15189 requires as well that for any laboratory test, so any laboratory examination method needs to be validated for their intended use. So therefore an intended use must be set up and defined for a laboratory examination as well. So what is the intended purpose about? The intended purpose is defined in the instructions for use of a device. So very often we do not have an instruction for use for a laboratory developed test. So this would be equivalent to your standard operating procedures in your laboratory that define how to perform an examination. The marketing material that you provide for your test services on your webpage, for instance, or any other print marketing material, for instance, the labeling of the device, but for a laboratory developed test, you may not have sufficient labeling information. But it can be any information that states a specific performance of your device, and statements which are to the benefit of an individual patient. These are all information that are part of the intended purpose and may define an intended purpose. So it's better to do it the other way around, to define an intended purpose, and derive the marketing material information from that. Before we can do that, you need to understand that the intended use is very often misunderstood from an intended purpose. So we need to differentiate between those two terms. So the intended purpose is defined by the medical indication, whereas the intended use includes as well transport, service, and maintenance, in addition to the medical indication to the intended purpose. So therefore, I recommend that you define an intended use document in your technical documentation, and this should define the medical purpose, the respective patient population which is targeted by the test, the intended use, so the scenarios that come along with it, and the users who are performing an action with the device during the examination. So the normal situation for a medical laboratory is that it is provided with CE marked IVD products from legal manufacturers. And in the case that a patient is consulted by a physician, a sample is sent to the laboratory, and the laboratory sends back a report after the examination. But what is very often happening is that the medical laboratory decides to change, to not apply the instructions for use of the manufacturer, to change the protocol, or to develop their own protocols. And in this case, where they develop their own protocols, it's very often the case that they use research use only products. And there you see that this is not intended by the manufacturer that these products are used for diagnostic purposes. So in this case, the medical laboratory takes over the full responsibility for the manufacturing of IVD products. So you see there will be no responsibility for the manufacturer with regard to IVD medical devices. This is fully transferred to the laboratory, and this is a heavy legal situation for the laboratory. To reach compliance of products, there is always a lot of documentation effort. You see, for research use only products, the efforts are very low for a manufacturer to place it on the market. And if laboratories buy these products, there is no reason why a manufacturer should take these efforts. There is another possibility. If the products are very generic, like DNA extraction kits, for instance, it could be possible that the manufacturers provide for general laboratory use reagents or kits. It may be true for software as well, but in this case, it's very complicated and to demonstrate the generic nature of the software. So to say, a pre-phase of or pre-status of IVD products is the so-called products for performance studies only, which is abbreviated with PSO here. These are products which already fulfill the general safety and performance requirements, but do not have yet a proven performance, a clinical performance. So therefore, they are used to assess the performance of the device before a manufacturer can place an IVD product onto the market. So there you see the documentation efforts and may understand why some manufacturers choose to provide only research use only products onto the market. With that, I would like to show you some examples for laboratory developed tests. So products or examinations in your laboratory fall under this definition if an in-house test is developed, so the so-called homebrew tests. If they use research use only labeled products, it would be a laboratory developed test, and a combination of different products processes could be a laboratory developed test if there are non-medical devices used. A misuse of a CE IVD, so if you use the product outside of the intended purpose or outside of the instructions for use of a manufacturer, it would be a laboratory developed test, and as well if you modify the CE IVD product. So what was the motivation of the legislators, of the European legislators to put the LDT scope into the IVDR? It was to the intention was to ensure the highest level of health protection. The rules governing in vitro diagnostic medical devices manufactured and used within a single health institution only should be clarified and strengthened. That use should be understood to include measurement and delivery of results. So you see, they wanted to put the LDTs onto an equal regulatory level as compared to the CE IVD products to make them somehow comparable. And the second motivation was health institutions should still have the possibility of manufacturing, modifying, and using of devices in-house, and thereby addressing on a non-industrial scale the specific needs of target patient groups, which cannot be met at the appropriate level of performance by an equivalent device available on the market. So this is heavily impacting the medical laboratories, since they need to prove that there is no device on the market with an equivalent performance. So, and therefore we find in Article 5 placing on the market and putting into service, with the exception of the relevant general safety and performance requirements set out in Annex 1, the requirements of this regulation shall not apply to devices manufactured and used only within health institutions established in the Union, provided that the following conditions are met, which I would like to demonstrate in the next slide. So that means products manufactured and used by EU health institutions, which means medical laboratories, must comply with the Annex 1 with general safety and performance requirements and need to fulfill the following requirements. It is not allowed to transfer the products to other entities, to other companies. You need to provide a proper quality management system for manufacturing. That means ISO 15189 is maybe not sufficient. So you need to at least apply some chapters of ISO 13485 to be compliant. The laboratory must anyways be accredited according to ISO 15189. And you need to demonstrate that the competitor CE IVD product is not does not provide the performance required for the specific patient group that you are targeting. So therefore you need to prove that yours is better suited to fulfill the specific patient requirements. A laboratory needs to provide information and justification to authorities regarding manufacturing, modification, and use of the device if requested by the authority. The laboratory needs to publish the name and address and of the laboratory and the identification of the products, and declare the conformity to Annex 1. And then they need to provide extensive documentation on manufacturing, design, performance, and compliance to Annex 1 for Class D products. However, it is also stated that member states may apply this provision as well to Class A, B, and C devices, which is true for Germany already. We know that this must be applied for all classes, but I don't know the situation in other member states in Europe currently. Products are manufactured in accordance with the documents that we just pointed out in under G. And the last point is the laboratories need to collect experience gained from the clinical use and apply, if necessary, corrective actions. So you see that somehow a summary of all the requirements for IVD products, for IVD medical devices. So the only very big difference is that laboratory developed tests are not under the supervision of notified bodies. So it's the laboratory's own responsibility to make sure that the tests comply with the IVDR and in particular the Annex 1. Yeah, with the currently compliant application of your laboratory developed test under IVD directive, the additional efforts to reach compliance with IVDR can easily be mastered until next year if you start from now to set up the technical documentation or the LDT documentation. But there's a but. In the case that there is a product on the market that meets the specific needs of the patient target group at the performance level of your LDT, it will not be allowed to further use the device after May 26, 2022. So this is a huge problem. As soon as you invest in your LDT, there may pop up an additional CE-IVD product which fulfills these performance requirements, and then you lose the option to sell your service after May 26, 2022. What is the alternative? Yeah, you can place the product onto the market or products that are used or developed in your own laboratory. And if you'd like to learn how to do that, you may visit our Ivdr Seminar, which we can provide in English as in-house seminar as well. And if there are enough requests, we can have an official seminar in English language as well. Thank you for your attention. This is what Jona Institute provides. If you would like to consult us for setting up a technical documentation or for strategic approaches, please consider us. Thank you.
Thank you, Sebastian, for this very nice overview and wrap-up of the requirements of the IVDR. And we already have some questions that have arrived, but we will address them in the Q&A session at the end. So let me check if I can, scroll to the next speaker first. So the next talk will be presented by Dr. Ben Liesfeld, who has obtained his PhD in physics, and after some work at a startup for ophthalmological medical devices, where he played a key role in the development of computer-guided laser surgery devices, he started working in a clinical laboratory for genetic diagnosis in 2013, where he was responsible for establishing IT, software development, and bioinformatics department. So this is where he learned about the huge challenges and questions that are in the field of particular NGS diagnostics. So this led him to found his own enterprise, and since 2014 he is the managing director of Limbus Medical Technologies. In his talk today, we will learn how to deal with software in clinical diagnostics, so software that is made for patients and not for research use only. So I'm handing over to you, Ben.
Thank you, Yvonne, for the kind introduction. And I'd like to take the opportunity here to shine a spotlight on the topic of software. And if you're working in the genetics space, you are probably dealing with a number of products that carry a big disclaimer for research use only. And I'd like to point out what actually has to be done to turn a device into a medical device that is really made to diagnose patients. Let's first revisit some details about the changes now that are coming up for software, and specifically Sebastian mentioned a few of them. I'm just highlighting them here again. In the old IVDD, software was not even considered as a standalone device. It was always part of a bigger system. While the general requirements applied, of course, also to software, but most of the other requirements were tailored to some big electronic or electric machines, basically. So the legislators now are considering software quite differently. They recognized that software can be an independent, standalone product in its own right, and they also recognize that it must be regulated as such, and there must be more emphasis on software as a standalone device. Again, the general requirements apply, but the IVDR now also mentions specific requirements for software. For example, state of the art of software development is mentioned, and also the software life cycle, which is a fixed term in the industry, is mentioned in the IVDR specifically. I'll explain in more detail here what these requirements actually mean for software development. Let's first make clear which kind of software, which software component in a laboratory should be considered a medical device. You may operate an instrument using software. There may be some device software on the instrument that operates it. You may analyze the data that an instrument generates with software that you install locally on computers in the laboratory, or you may utilize a cloud service provider like we are to perform data analysis. One very important aspect, one criterion to determine if software is a medical device is, for example, if the results from the software could also be would be comprehensible without using the software. Meaning, could you take a piece of paper and a pencil and come up with the same result? That's kind of the criterion. If it's not the case, if the calculation or the analysis is opaque to the user, then it's probably going to be a medical device. The only kind of software that's exempt from regulation is and will still be laboratory information systems, because they are basically considered to be typewriters. They are only storing or displaying information or performing very basic calculations. So laboratory information system manufacturers can still relax, but basically any other kind of software will have to be examined very closely. We are asked quite frequently by our customers, well, I'm using Excel in my laboratory process. Would Excel become a medical device? I mean, I'm just looking at data, I'm reviewing data, I'm not modifying it. I'm not Excel is not a programming tool in my purpose. I'd like to mention that there's a quite famous study on Excel attachments in genomic journals, and these people have performed a study determining if there are errors in those Excel sheets, and they found 20% of all Excel attachments, they found errors in gene symbols. This is basically very popular or a very annoying problem that geneticists have when using Excel. And what is the solution that we have in the year 2020 for this problem? Well, HDNC decided to rename genes. For example, SEP2, the gene SEP2 is now called Septine2. So just for the purpose that Excel does not convert the string SEP2 into the date September 2nd. Is this a good solution? Well, maybe for the future, but still we have a lot of historic data that are analyzed in Excel, and we are going to continue to have these issues. So Excel is a software, and modifies the information that you're looking at without you even knowing or you even being able to control it. So let's take a look at how would software be classified in genetic diagnostics. Classification is a means to determine if a product is, for example, of high risk for patients or of low risk, and how you have to deal with it in the regulatory process. According to IVDD, classification of software was very simple. Well, there were no requirements specifically for software and no classification for it, so it was simply other. It didn't fit into any of those specified categories, so it just was subsumized under with everything else, basically. IVDR now changes this dramatically. And for example, for our software, which is used in genetic testing, there are at least 3 items in Rule 3 of Annex 8 that specifically state that our software product will be a Class C device because it's being used in cancer diagnostics, for example, or in human genetic testing in general. So there are genetic testing has become very important, or has it become a very important item in the IVDR. If we are now taking a closer look at the requirements that I mentioned earlier that have now been, where the bar has risen a lot for software, there is, of course, the Annex 1 that states the general requirements for IVDR products. And as Sebastian already mentioned, these general requirements apply to all medical devices, to all IVD, no matter if they're used in-house and just laboratory developed tests, or if they are properly marketed medical devices. You can't get around those general requirements. They comprise, on one hand, requirements regarding IVD, like we know from the IVDD regarding performance, reproducibility, reliability. Of course, that's basic requirements that you have to demonstrate. Anyhow, software now is addressed with very specific requirements. You have to comply with the state of the art. You have to have processes concerning the life cycle of software. You have to perform verification and validation, perform risk management, and also IT security is mentioned. So these are terms that are well known in software engineering. So the IVDR now includes very important provisions that have been, this has been necessary for a long time. It has been neglected in IVDD, and actually it's about time that this is now included in the new IVDR. For a layperson, somebody who's not dealing with software development, these may seem like very general terms and may not be clear what is meant by them. And you may ask, well, is it not possible to simply put software on a test stand and demonstrate that it complies with IVDR or with regulation that is of good quality? Well, let's have a look at the question, can you examine and inspect software? And I like to use a picture here, or an event that happened in February 2021, where an accused murderer won the right to actually check the source code of a piece of software that was used to generate evidence against him. And the manufacturer of the software was trying to fend this off for quite some time. Finally, the manufacturer lost, but his main argument was that the review of 170,000 lines of MATLAB code that his product comprised would take more than 8 years. Well, of course, the manufacturer was not willing to have this review being performed, but this statement gives you some idea that it seems to be complicated or difficult to review software. And if you compare software to something that you know from your everyday life, and if you're asking, how easily can you read it? If you compare it to a piece of world literature like War and Peace from Tolstoy, more than 1,000 pages, about 73,000 lines of text, you may say, well, it's quite difficult to read this big book, but there is one convention that makes it actually very easy for you to read it. You start at the first page and you go page by page until you're at the end. Then you have read it and probably also understood everything that's in it. But with software, this is quite different. For example, our software contains more than 350,000 lines, so it's probably much more comprehensive than War and Peace. But what makes reading it very difficult is sections of the software code depend on each other. It's not executed in a linear way. So in the beginning, it may not even clear where to start. So software can be a very complex thing and cannot be easily reviewed. So you need to have other means to actually demonstrate that software is of good quality. And this is when the term life cycle of software comes in. Actually, the only way to prove that software is of good quality is to document that you developed it in the right way. The life cycle of software typically starts with specification. You specify what the software should do. You implement this into source code or into software. You perform verification and validation in order to demonstrate that your specifications are met. And then you can release the software and hand it over to users. But your responsibility doesn't end here. You need to perform surveillance. You need to observe if there are any issues in the field when your customers, when your users are using your software. There are specific requirements for the kind of processes that you have to implement. One core process is risk management. The risk for the patient is a key idea, a common thread that runs through both MDR and IVDR, through all the regulations. Everything circles around the risk for the patient. You need a proper process to manage that. Usability has become increasingly important as well, because we recognize that operating errors originate mostly from bad design. It's not the issue, it's not the fault of the user, it's rather or they can be attributed mostly to design flaws. You also need to perform a proper management of configuration, of changes of your software, and you need to be able to handle problem solving in a transparent way. The good news is for all of these processes, there are recognized international standards that you can follow. ISO 14971 for risk management. There's a standard for usability assessment, and there are more general standards that tell you how to perform these other processes. So from our point of view, since software is never really finished, after the release is basically before the release, you're undergoing these processes all over again. And for the example of our software, we're releasing new software versions 4 times a year, so we're practicing this quite a lot. Let's explain a little bit more with our software as an example what verification and validation V&V comprises. If you look at our software consists of more than 10 programming languages. 65% of those languages are of source code are written in Java, 30% in TypeScript. The kind of technology that you use, that you decide to use in your medical device determines significantly how easily you can perform V&V. And as I mentioned before, it has grown to a high degree of complexity with 350,000 lines of source code. So how do you test this big piece of software? Well, first of all, you need to break it down into small units, and you basically test every single unit if it performs well. So you give a function a certain value, and you inspect if the return value is the expected result. Then you put those small units together to larger units, to modules or components. You perform integration, and you perform integration testing. You test those modules, and in the end, you perform something that we call end-to-end testing. It's basically simulating how a human being would use your software, the entire system, and you demonstrate that the software behaves as expected. A frequent question is, well, how much effort do I need to put into this? I mean, testing, this can be exhausting. What is expected from a medical device manufacturer? What is the state of the art? From our point of view, it is mandatory that testing is automated. If you do not automate software testing, it's always an additional effort to do that, and you probably tend to do less testing that you actually should do. So by automating this becomes something this becomes integral part of your development process. In our case, 30% of our software code is actually software that tests our software. So we invest a significant amount of our time into creating software for testing purposes. And we also execute this very frequently. We spend more than 5,700 CPU hours every month for test execution. And we also try to get better every time. So I look back at the numbers from 2019. Back then, we had 4,400 automated tests being performed on our software. And we were able to increase this number to more than 7,000 individual tests that are performed before we release a piece of software. Now, a question that many people ask who have created their own software solution. And in genetics, that's very common because 5 years ago, there was no well-established software solution, not even near being compliant to medical device regulations. I'd like to use two images here to highlight how difficult I think this is. So I'm putting together two quite different vehicles here, and it's not the point that the car on the left side is old and well, colorful. On the right side it's a new car, or it's electric versus fossil fuels. It's rather the fact that the car or the device on the left has been customized very much to the needs of the user. It has been modified many times. It works very well, but if you consider this, being a piece of software, it is now very difficult to actually obtain to document that it's still compliant with regulations. You just can't put it on a test stand and test lighting and exhaust and brakes. It's not impossible. It's just impossible. The only thing that you can do is take it apart entirely and put it together piece by piece, and that process document how you did it. And this, in the world of software, means basically develop the software from scratch anew. So it's a big effort. So I have basically two short take-home messages for you that you're working in the laboratory space or even in genetics. The first one is, do not underestimate the state of the art. When the IVDR mentions state of the art in the software world, it means really very specific things, and there are recognized standards who tell you how to do it. And the second message is, well, choose a future-proof solution. As Sebastian mentioned, IVDR is going to be enforced from May next year, and there will be no delay, and it is going to stay for the next 20 or 30 years, like IVDD has been. So it will determine the way that we do business in Europe, and we know what to do to be compliant, so we need to get at it. Thank you.
Thank you, Ben, for your presentation. I would just want to ask you the first question that is just arrived regarding the state of the art. So what does this term, the generally acknowledged state of the art in the field of medicine, mean in particular?
Well, I can make a statement about this from the point of view of software. State of the art is typically referred to, what is recognized in standards. It's not just what is informal way of doing things in the field. Typically, if you're talking to a notified body who's reviewing your processes or your devices, the good point of reference is international standards. That's at least for you or for the manufacturer, for the developer, this is a good way to demonstrate that you apply state of the art.
Okay, thank you for the answer. Whoops. Then we will turn to the third speaker today, Björn Nowak from SenseID, giving a sum up of the performance evaluation, what it means and how to deal with it. So Björn, welcome today. Glad to have you on board. Björn is a biochemist by training and has a professional background in the research and management with a focus on liquid biopsy. He has many years of experience in pharmaceutical and life science companies working according to ISO and GMP standards, and since 2017 he is the co-founder and CEO of SenseID, a company that develops, manufactures and markets reference material and quality controls in DNA diagnostics. So today we will hear more about performance evaluation, and I'm handing over to you, Björn.
Thank you, Yvonne. Okay, that works. Welcome everybody, and thanks for watching. Due to the sheer size of the IVDR, I can only discuss a small part of the performance evaluation here. But however, we are of course available for further questions even beyond the discussion round later on. So I really hope this short introduction to the performance evaluation will help to get some first insights on how to deal with it. First, let me make a really short introduction about SenseID. We are, as Yvonne already told, developer and manufacturer of clinical genomics quality controls and reference materials, which are in regulatory and customer compliance. So what that means, we have a management system that is certified according to ISO 13485. And we do have the ability to provide CE, IVD, quality controls that could be, as we do have OEM and licensing options as well, could be included in your assays, or could be used as well for performance evaluation or for research use only, and so on. And we do have a lot of possibilities to customize the products, and we typically say that we make the products fit for purpose, so fit for the need that you have, which means it could be customization of allele frequency in genomic DNA, cfDNA, ctDNA, plasma products, or even formalin-fixed, paraffin-embedded sections. We can customize concentrations, mutant copies, filling volumes, so basically any kind. The content of the webinar will be about, first thing is about the classification and requirements for genetic tests according to the IVDR. Second will be performance evaluation and application examples. And third part will be a short summary, actually only on one slide. So let me start with the aim of the IVDR. And that is, as Sebastian already explained, to ensure a high level of safety and performance throughout the life cycle of a genetic test. So for your laboratory developed test or the CE IVD labeled genetic test, the following applies. It's namely rule number 3 and 4, and there you can see that devices are classified as class C if they are intended for specified purposes, like for infection, diagnostic, of course not all, but to be used as companion diagnostic and many other things. By the way, as this is the current situation and was already a question, devices intended for self-testing, like SARS-CoV-2 self-tests, are also classified under the IVDR as a Class C device. So whenever your device is classified as a Class C device, you have to meet specific requirements, which means in this case a quality management system according to Article 10 and the use of harmonized standards, like for example ISO 13485. Safety and performance requirements and technical documentation, as well as conformity assessment procedures according to Annex 9. And that means quality management plus assessment of technical documentation, including testing by EU reference laboratories. So the question that we are confronted with more and more is, do I have to, or do you have to, as an LDT developer or IVD developer, have to do performance evaluation? Performance evaluation is specified in the IVDR. And the simple question, we can ask two different questions here, actually, because IVD manufacturers or LDT manufacturers, do you have to perform a performance evaluation? And the simple answer for IVD manufacturers, yes, of course. This is according to IVDR Article 56 and Annex 1. And the second question would be if LDT manufacturers also have to do performance evaluations. And also here the simple answer is yes, but this time according to IVD Article 5, Paragraph 5 and Annex 1. So here are the performance characteristics at a glance: clinical performance, analytical performance, and scientific validity. These taken together are meant to be the performance evaluation. And so IVDs and LDTs must be designed and manufactured in such a way that they are suitable for the purposes stated by the manufacturer, and in terms of performance taking into account the generally recognized state of art, which Ben already explained a little bit what that means. The performance characteristics of the product shall also be maintained during the lifetime of the product as specified by the manufacturer. And the performance evaluation is part of the technical documentation. So this has to be considered as well. So let us have a closer look on scientific validity. Also here, IVDR explains the terms. And the question is how to show evidence of scientific validity. And this is also explained by using any of the following sources or combinations of these sources: relevant information on the scientific validity of devices measuring the same analyte or marker, scientific literature, consensus expert opinions, positions from relevant professional associations, results from proof of concept studies, results from clinical performance studies. So how do you demonstrate scientific validity? Well, you should start actually from the beginning with a start of the development of the test. And you should systematically document the research and the literature, which should be at least peer reviewed. So as an example, question here, what if the literature is insufficient or the test is based on barely described novel analytes or markers because you're introducing something really new to the market? Well, then you have to do or have to conduct feasibility studies to demonstrate clinical and analytical performance characteristics. So the next performance characteristic is the analytical performance, which is also specified as as it means the ability of a device to correctly detect or measure a particular analyte, and how to show that how to show evidence of analytical performance means in relation to all the parameters described in Annex 1, Section 9.1a. And there it is described that you have to show analytical sensitivity, analytical specificity, as well as trueness, precision, accuracy, limits of detection and quantification, measuring range, linearity, and cutoff, including the determination of appropriate criteria for specimen collection and handling, and control of non-relevant endogenous and exogenous interference cross-reactions. That is a lot. We know that. So how do we demonstrate analytical performance? You can do that as a part of verification via performance studies. And our tip would be to carry out analytical performance evaluation as a complete test, which means you should start at sampling, do sample handling next, and have to do validated measurement, use validated measurement methods. If possible, you should try to find metrologically traceable controls and afterwards do the evaluation. You should also compare with known reference methods and or reference standards. So as an example question here, again, the test or your test could probably include novel markers like mutations without known references. What can you do? Well, the sample material used for studies must be very well characterized. So for example, you could use certified biobanks which probably have patient material available, or you can also use certified reference materials, which could mean two things. One would be a CE IVD labeled reference material, or it could be certified reference materials. Certified reference materials are not really often available for genetic tests nowadays, but CE IVD labeled controls can be provided, for example, by SenseID. So if no reference material is available, what then? Or no valid reference methods are available, then the performance characteristic accuracy of your test is not verifiable. So the next performance characteristic is the clinical performance, and that is also defined in the IVDR, but I will not read it now, as you can look it up later on. And how the question is how to show the evidence of clinical performance. You can do that in relation to all the parameters described in Annex 1, Section 9.1b in this time. It is diagnostic sensitivity, diagnostic specificity, the positive predictive value and the negative predictive value, likelihood ratio, expected values in normal and affected populations. Populations. So how do I or how do you demonstrate clinical performance? Well, you should do that, of course, through clinical performance studies. And in this, clinical performance equals the diagnostic accuracy. You should plan and prepare a report in accordance, for example, no, not for example, in accordance with Annex 13, Section 2 and ISO 2916. You have to evaluate all parameters according to Annex 1. And as an example question, could clinical performance studies also be waived, which would be good? And the simple answer here is yes, but only if established and standardized tests that are equivalent to yours are already available. Sufficient data is available that relate to routine diagnostic tests. But caution, if an LDT is to be used despite an existing CE IVD test, then it must be demonstrated that this LDT corresponds to the same performance level associated with the same intended use of the commercial test, which was already explained a little bit by Sebastian. Yeah. So our tip would be make a cost or and benefit analysis in advance, because it could be really costly to perform all of this. So what else? You have to show stability. This this also includes evidence that the device is stable. So here the following characteristics are to be demonstrated in studies: the claimed shelf life, the in-use stability, and the shipping stability. And guidance on how to do it can be found here, for example, in the EP25 evaluation of stability of in vitro diagnostic reagents and in the ISO 23640. So you complete the performance evaluation with the post-market performance follow-up. And as all performance evaluation activities are part of the entire life cycle of the product, you have to do the evaluation activities again and again and again. Performance evaluation is therefore an ongoing process, and manufacturers are obliged to update the performance evaluation data through this continuous monitoring. Here we have two examples. So example A would be your genetic test, for example, examines just 5 mutations that are relevant for to the therapy, and new findings add 3 more mutations. Then you are now required to redo your analytical and clinical performance evaluation for your test and reevaluate all performance characteristics. Yeah, by this you can imagine that it's really hard to find the necessary patient materials, for example, but for that reason, companies like SenseID exist and can provide reference materials that can help you to evaluate that. Example B would be the accuracy of your test no longer corresponds to the analytical state of the art. Then you need to prove the analytical performance by new studies, taking into account new methods. So with this, I come to the summary. And if you are a manufacturer of a genetic test, an IVD or LDT, then you should carry out performance evaluation. You should follow ISO 29916 in planning and implementation of this, and create a performance evaluation file. We should implement the performance evaluation as an ongoing process, post-market or post-marketing performance follow-up. And if there is a comparable product on the market with a CE IVD label instead of the LDT, then this must be used, or the LDT needs to be validated against it. So for LDT manufacturers, the IVDR or MDR, if applicable, and thus the implementation of the requirements of a QM system apply. And of course, also more or other national directives or regulations and laws may also have to be complied with. Like in Germany, for example, the Rillibeck. So with this, I would like to thank you for your attention, and I think all of us are open for questions. You're welcome.
Thank you for your presentation, and welcome now everybody back to the Q&A session. So we have all three speakers here, and we also like to welcome Andreas Wollmann to the Q&A session, who is the head of regulatory affairs at SensID and is also here to answer questions. So quite a few questions have already arrived. I would just pick some here. One would be, when IVDR harmonized standards are expected to be released?
Oh, that's a difficult question. The harmonization approach is already initiated, but I cannot state when this is to be expected. We hope that they progress fast, but I don't recall a particular deadline that is mentioned in the implementation that was set out by the European Commission.
But this is certainly a topic that concerns all medical device manufacturers, so we are really waiting for this to happen. And it's a difficult situation. Yeah, so from my point of view as a medical device manufacturer, also talk to your notified body about the best way to handle this. And yeah, you have to document. If you're choosing a specific standard who's not harmonized, you're allowed to do so as a manufacturer, but document. I can recommend to document how why you are doing it.
Fully agree with that.
Okay. Then there's a question, does the term no transfer allowed also apply to laboratories that have multiple labs across Europe?
If they're headed under one legal entity, I don't have concerns about it. But if it consists of several legal entities, you should find a lawyer who is, so to say, representing your opinion on that, and gives you a way to argue for it, that it still is possible to apply it. There is a huge discussion out there, and currently we don't have cases where courts have made clear-cut decisions.
Okay, then we have a question that is like a more complex question. A laboratory uses an LDT based on PCR with an intended purpose XY. A commercial PCR test with XY purpose does not exist, but a commercial IVD product based on mass spectroscopy with exactly the same purpose exists. Am I still allowed to use my product, or do I have to switch to the different technology?
This answer is really difficult to answer. Yes, the question is difficult to answer. It depends very much on the specific patient needs. So if there is a specific variant or mutation that can only be targeted by your PCR test or a performance aspect, like the sensitivity is much higher of your PCR test as compared to the mass spectrometry technology. You may place your product specifically to not yet pre-diagnosed patients, or to use it as a screening test, whereas the other testing could be rather conformative, could have a conformative purpose. If you place it somehow differently, you may find your niche. But that's important to really argue for, to find arguments that a specific patient group is targeted. Even the price may be a decision point. If your test is much cheaper and it's not yet reimbursed by the insurance system, it may be a possibility to state with the lower price you reach a target population that cannot afford this expensive testing. So there are a lot of arguments you can choose.
Yes, and so my recommendation, the performance evaluation process is mandatory for all products with a diagnostic purpose. So,
Okay. One more question. What does it mean? Does it mean that as soon as there's a new product, CI certified coming up in the future, are we obliged to use it even if our LDT was already in place before the commercial kit came to the market?
Yes, that's true.
Short answer,
So,
yeah.
bit more answer to that question. There are a few options you can take. I mean, you can change your intended purpose. As I said before, you have some arguments and some directions to go for. It's a strategic approach, or you can already by now start to document according to the IVDR requirements so that you have a technical documentation in place, and then it's just a matter of supervision by a notified body. So you engage with a notified body, put your CE mark on, and sell your test. And if you would like to go a more progressive strategy, then start being compliant from the beginning on. So start directly and have already the plan to put the product onto the market. If you see a market for your product, it could be an option to sell it as a CE-IVD product as well.
To what extent would the mandatory documentation of LDT make internal trade secrets public, or is the documentation only made available to the regulating bodies?
IP is yours. It should be. I mean, the European Union has very much interest that your rights are conserved in your company. Only if you consider to license your development to a manufacturer, you need to provide all details because the technical documentation must be available at the site of the legal manufacturer. So the one who is stated on the labeling of a CE-IVD device as legal manufacturer must have access to the full technical documentation, and that means all technical details, as Ben has described, all those technical details regarding units of a software and all the technical details like sequences of primers and probes should be available at the legal manufacturer sites and not at an original equipment manufacturer only. So take care of this.
How will regulatory monitoring of laboratories developed tests look like under the IVDR?
Yeah, that's a good question. But so there are, of course, there are authorities that are relevant in IVDR. The qualified authorities, for example, in our case, this is a state authority in the state of Mecklenburg-Vorpommern here in Germany. So it's different for every European country, but they are mentioned in the IVDR. But maybe Sebastian, you could tell a little more details how active they are, how active they will regulate LDTs. Have you any ideas about that?
I do have an idea, but I cannot provide evidence for the future or what the future situation will look like. So currently we face that we have requests for training of competent authorities. So they get trained more and more specifically for the purpose of software surveillance. So they will be able to judge, but you need to take into account they are not most of the time they're not software experts, because the software experts are needed in other roles. So therefore, really take care and be prepared to a situation where a competent authority will visit you. Currently, we don't see that they are increasing the headcount within the competent authority bodies. So I don't expect more supervision in terms of more frequent visits at laboratory sites. But this is only a view on Germany. I cannot state what the situation is like in other countries, and that's in the authority of the member states.
Yeah, we have a software question. What about open source software? When it was developed by another lab and it works well, can I still use it?
Well, it must be, it doesn't matter what the license model is for the software, if it's open source, if it's closed source, it has to fulfill the same requirements. It doesn't make it better if it's open source. It just needs to comply with the same state-of-the-art requirements as mentioned in the IVDR. So treat it as maybe as a software of unknown provenance in your case. So there are specific provisions in the international standards how to treat such third-party components in your own, probably in your own solution.
The benefit of open source software is that the source code is readable and understandable. If it's a small component, you may take the chance to read the code and understand the code. However, the problem is if you cannot access the code bases, then you have sort of black box testing for these soup units. Therefore, you should have a sophisticated testing strategy so that you're sure you're always you have the power to judge whether the software of unknown provenance does what it should do, and you should observe the changes or the so to say, the websites that show whether the software behaves as unexpected, to judge whether this has impact on your diagnostic statements you make with your software.
One question, how can sufficient clinical samples be obtained in the context of rare diseases?
Yeah, you have to, as explained, you have to go to biobanks, ask them if they have sufficient material available for rare diseases, for example, but you can also ask biobanks to source these kind of samples. That would be the best way, because then you can have access to patient samples. However, if these are not available, you can also source control material from companies like SenseID, and these can be provided, tailored to your needs. Especially in when you're looking for example, plasma samples, and that are typically not easily available from patients in a sufficient amount, then we have the possibility to supply these kind of samples with specified allele frequency, concentration of DNA, so you can also show limit of detection, limit of blank, or limit of quantification of your test.
So if a CE IVD genetic test is updated, it includes now a new gene or a new genetic variant for the same condition, is it necessary to certify it again?
Well, that's a good question. It depends on the status of the database. So there's an FDA guidance document out there providing some insights how a database should be handled. So it's if you're referring to publicly accessible databases which make the connection from the variant to the scientific validity, so to the clinical picture, it is possible to so to say, make the supplier qualification of that database maintainer provider. So this is the basis of this FDA guidance document. So that could be a possibility to routinely release the new versions of those databases into your softwares. So you shouldn't directly access or let your software access to those databases, so that that you have the control over the knowledge base of your software. That would be my answer. Yeah.
I have understood the question slightly differently. Is it
Okay.
change management in a, for example, an NGS panel, certified NGS panel?
It was not specified.
Okay, so
okay, let's take the chance to answer that as well.
Yeah, so let's just so if it was about change management for a CE approved test, of course there are rules to deal with that. So of course a change it depends on what the change is. Addition of a gene to a specific test, so expanding the coverage, the purpose of the test, even modifying the intended purpose. This certainly triggers some activities with respect to the notified body. So if you're a manufacturer of a CE labeled IVD device, you have a notified body anyhow in the genetic space, and you need to consult with them, and they will also tell you which kind of changes will trigger an additional review. So if the changes are small and do not affect intended purpose, typically they do not require an additional review. If the changes are significant, then the entire process starts again.
So I fully agree. It's heavily in discussion currently because under IVD directive and medical device directive, there were guidances with respect to significant changes that should be submitted to the notified body in the situation that you are under the supervision of a notified body. So that would be applicable for class B, C, and D devices. But currently, we see that IVDR opens up the possibility to have more significant changes that are not that don't need to be submitted to the notified body. That is for standard class C and B devices, because it is dependent on whether the notified body issues an EU certificate, and this seems to be only applicable for CDX, for point of care testing and for self-testing, and for class D devices every time. It seems to be the case, but this is not yet clear whether there will be guidances speaking against it. It seems to be the case that if your product falls into a generic device group, that it's possible to have changes under that certificate without notifying or without submitting those significant changes to the notified body. But this is take this information carefully. You always need to consider whether the notified body certification rules are different to that or have requirements in addition.
Yes, so question regarding a corner case. So do private companies providing a clinical test need to comply to IVDR? The test is not sold as a reagent or kit, but is provided as a service.
Service is to be regarded equally. If you look into ISO 13485, it clearly states product means as well service. So in the case that you provide service, you may even do more because you need to fulfill the laboratory requirements as well. So ISO 15189 is an additional requirement, or in Germany, you need to apply the Riliebeck for quality assurance in medical laboratories. So therefore, take care. This is a dual aspect which needs to fulfill even more requirements.
And we have one question regarding the US. So the IVDR explicitly state for LDTs in EU member states. What about clinical trials where EU samples are tested for example, in the US? Are LDTs designed and validated by diagnostic laboratories for patients' exclusion and inclusion? Will these tests be subject to the IVDR? And if there's an equivalent to a device available, must it be used in the US to test EU patient samples?
Again, a very interesting question. The simple answer is yes. That's I don't know the English term for it, but it's called Fernabsatz. It's Article 6, if I recall correctly, where a service is provided outside of the European Union that is directed to a legal person which is inhabitant of the European Union, the IVDR must be applied. The difficulty is that you will not find a CE IVD labeled product in laboratories in the US or anywhere else in the world outside of the EU. So it will be very difficult to demonstrate the compliance of IVDR outside the EU for a product applied outside the EU in a medical laboratory. So we don't know how this is ruled out in reality, but yes, the simple answer is yes, it must be applied.
So if the if my CE IVD isn't approved by May next year, but submitted to a notified body, is it still acceptable to perform testing on the CE IVDD assays that were manufactured prior to this to the 26th of May next year, or are there any caveats to this? Did you understand the question?
Could you repeat the question, because I did not
I think it's
recall.
about can an IVDD device be used beyond May 26th? It's planned that it is being submitted to IVDR review, but it has not been approved yet. Can you continue to use IVDD devices?
The important thing is that the IVDD compliant medical device needs to be placed onto the market before May 26, 2022. That means the ownership must have changed from the manufacturer to a distributor or to any other legal entity. In that case, it is possible to make it available and install it at the customer side for further years until May 2025. So 26th of May 2025 is the final date where no IVDD compliant product can be installed or made available anymore. For an instrument, that means it can still be applied in a medical laboratory beyond that date of 2025, because the life cycle of the product can be even longer. But it means selling those who are at the distributor side in stock is still possible, dependent on the shelf life of the product. So it will be very much limited by the shelf life in case of reagents, but software will be difficult because software is subject to frequent changes. But for instruments, it may be even possible to sell and use them beyond the application date of IVDR.
The important thing is changes to the IVDD devices will be limited, right, after May 26th.
Exactly.
So they're basically in a frozen state, and in a market like genetics where products change frequently, this is an important restriction.
Thank you for the addition.
Okay, one last question. How do companies, how will they know if a new CE IVD test is in the market when they are using their own test and it has been okay so far, but now there's a new product? How will they know?
The essential requirement is that post-market surveillance needs to be performed proactively. So that means you're in this responsibility to observe the market, whether there is an equivalent one available.
Okay, so we're not able to address all questions, but we will follow up with you and send you the answers you need afterwards via email. I thank all speakers again for sharing their expertise, for answering all the questions. Thank you all for joining. And if you are interested to join upcoming varvis webinars, you can sign up for a newsletter and then we keep you in the loop. Until then, stay safe, everyone, and goodbye.
Thanks for watching. Bye.
Thank you very much, Limbus.
Thank you very much. Bye-bye.
Bye.